Amonida Zadissa from the UK. Actually, Amonida is a director of informatics from UK Dementia Research Institute, and she has been an associate for 2.5 years, yes, as a senior strategy officer at European Bioinformatics Institute of European Molecular Biology Laboratory. I guess a lot of people are familiar with that. It's called EMBL, EBI. I know some of us are familiar with that particular organization, and it's a very big organization. Sure, we have a very big person in our midst today. And she actually owed an MSc degree in molecular biotechnology from Uppsala University, and she has a PhD in bioinformatics from Otago University. And she has worked within human data space for over 15 years, and that includes 11 years in example projects as a bioinformatician, annotation manager, and a coordinator. So we have an amazing person in the house. So Amonida, it's nice to have you on our platform today. Thanks for honoring our call.
Thank you so much, Udon and Toby for inviting me. The honor is literally mine, and it's great to be here. Do you want me to, shall I share my screen?
Please, you can do that. Let me stop sharing.
Thank you. So hopefully you will see my screen now.
Yes, perfect.
Excellent. Okay. So again, thanks very much for giving me this opportunity. I really feel privileged to be here, and I hope that what I'm going to tell you about will in some way, support you, help you, or in making your decisions when it comes to bioinformatics. So I call this my talk From Genomics to Dementia, my data-driven journey. Super geeky, but I hope you will you will follow me through this talk, and hopefully it will become a bit more clear as well. So this is a map of the world, as you can see. A bit different to how we usually see it. That's why I specifically chose this map, because I think it's actually more representative of how the world should look like. Anyway, so this is, let me see. Okay, so this is where I was born. I'm actually originally from Iran. I was born very close to Caspian Sea in the north part of Iran. And that's kind of, I guess, having two very technical parents in mindset, but none of them having university degrees. They were very keen for me to take that particular cassette player that you see apart and put it back together a few times, even though I was not really successful in making it working yet. But that kind of really started my interest in maths and physics. So I, together with my family in my late teens, moved to Sweden as a refugee from Iran. And that was a journey by itself, which still I am quite mindful of that as well. And while I was in Sweden, as Udun mentioned, I decided that I actually wanted to enter into the world of biology, even though I had avoided it while I was in high school. So I did a master's in molecular biotechnology, and that was where I actually the first computer, and did some gel electrophoresis that some of you, or perhaps all of you, are familiar with. And now equipped with an EU passport and the privilege and security that it comes with, I decided that I actually want to venture out of Europe, and I decided to go to New Zealand. I had a fantastic lecturer in genetics, and I really wanted to actually do this journey, both in terms of moving into genetics, but also physically moving out of Sweden and exploring the world. And while I was at the Otago University, I did my PhD in bioinformatics. That was at the same time when the human genome was coming out. It had literally been sequenced and published, and it was an amazing era to kind of be a bioinformatician. Because everything was new, as you can imagine. The human genome was out, and everybody was trying to figure out what this was going on. And there were all these terminologies that we used back then, which now we don't. For example, junk DNA. We don't call anything junk anymore because we realize that every part of the genome actually has got a functionality. That was also the time where I ventured into Perl. That was the language to learn if you were a bioinformatician. And I taught myself how to code in Perl, because before I was actually taught C++ and MATLAB. And again, equipped with a PhD in bioinformatics, I thought I could solve everything, obviously. So I moved back to Europe and to the EMBL EBI, so the European Molecular Biology Laboratory, European Bioinformatics Institute, which is based just south of Cambridge, just north of London. Excuse me. And during my time there, and also at the Sanger Institute, I learned how to annotate genomes, look at the quality of data, doing data analysis. I got involved with strategy and COVID head as well. So I was quite involved with all of those. And then staying in the island of UK, I moved to the UK Dementia Research Institute, where I am at the moment. And here I look after the data infrastructure for the whole organization. So I will take you through some, kind of touch base on some of the aspects of this journey up to now. And hopefully this kind of circle will show where we are on this journey. So we start with genomics and the Ensemble project that I was part of. As I mentioned, I was part of the genome annotation team, where we had a set of standard pipelines for annotating genomes. We would download assemblies and basically work with them, make sense out of them, and provide it to the wider world. All for free, because the whole idea of Ensemble is open science. While I was doing this work, I also got involved because I guess I was kind of more interested in data quality, and I also took on more responsibilities. So I became responsible for the human annotation, where we pulled together automatic annotation that our team was doing with manual annotation, which our collaborators, also basically based next door, were doing together. And for doing that, it meant that we had to kind of merge these different structures together automatically. And the result of that was, developing a pipeline called Havana Ensemble Merge Pipeline. Now we are talking about 20 years ago, when the stuff that I'm mentioning now. So all of this has changed now. So if you go to the Ensemble website, you will see reminiscence of what I'm mentioning, but the concept's still there. There's still a lot of merging and kind of combining different kinds of annotations together. So this kind of merged approach that we had was specifically made for our animal models, like the top models that we use. And those were, at the time, human. Yes, we're also animals. Let's not forget that. A mouse and zebrafish. And while I was doing this, what I noticed was that there were a lot of duplicated transcripts, which of course caused a lot of, increased our false positive rate. There were a lot of inconsistencies when it came to exon merging. So, and I was working with existing code. So I thought, okay, no, let's refactor this. Let's kind of take this apart, pull different parts of it together, move things around. But then after doing some work, I realized that actually, no, I need to rewrite this entire code from the beginning just to make it sense. On top of this, we also had live updates that were being made to annotations. And if any of you have worked, not if, I should say, all of you know that when, as a bioinformatician, when you're working with data, when you're actually developing your code, you really don't want your data set to be changing under your feet. You want it to be stable so you can actually look at it and confirm and actually be confident in your results. So this was very, very difficult. So what this also meant that was, so I introduced as part of this whole process, when we were kind of putting together automatic and manual annotation, what was to introduce data freezes, which allowed the teams to work to a certain deadline, and then they will let the, ask the people working on the automated side of things to actually basically upload the data and make sense out of that. All of this work led to the GenCode publications of the human and mouse genome annotations. And I'm sure you're all aware the GenCode gene sets are basically the reference gene sets, which are also part of the ENCODE project. And during this time, I worked with amazing set of people, I have to say, to start with, and also beautiful species. And all of them, most of them were in the category of small and cute, or in the category of big and ugly. And there was only human that was in the category of big and ugly, I have to say. Everything else was small and cute. So that was kind of, that's like I guess my really bioinformatician heavy journey, right? And so during my time at Ensemble, the things that I learned were, how do you deal with assembly qualities? How do you look at repeats? How do you, as a bioinformatician, how do you deal with telomeres and centromeres? How do you work with existing code? Well, in my case, I don't know how to rewrite, but Ensemble project was very large and still a very large project. And you do enter kind of midway into this journey. And how can you contribute to making this project even better? I became a pretty good wizard at shell scripting, I have to say, because I realized there is a lot of things that you can do on the command line with just shell, and that was amazing. I also learned, perhaps inadvertently, how I needed to actually respond to deadlines. And it was quite exhilarating, but also challenging path, because deadlines were part of the whole release cycle of Ensemble dataset. I learned how to prioritize, do you redo the entire genome annotation because perhaps 10 of your transcripts are incorrect? Or do you actually go, I go with the 99% of the genes being fine? I also learned how to manage a team of bioinformaticians. That was when I became the annotation manager, and working along with them together for all of us to kind of realize the pipelines that we're working on, how we can improve them. But I guess the main thing I learned was that it's absolutely fine to make mistakes. It's, I don't know how many times I made mistakes. I have no idea how many transcripts I put out which were incorrect, but that's absolutely fine, because this is a work in progress. There is nothing that is perfect in this world. We're just aiming to improve ourselves, and it is absolutely fine to make mistakes. Now that was it, like the start of my transitioning out of a bioinformatician role as in a hands-on coder into more coordination and strategy roles. And this was literally just before the COVID-19 pandemic happened. And I'm just going to touch base on what my involvement in the project, which was the European COVID-19 data platform, and what I learned during this project. So the whole idea of the platform was that we will take all the data, publish data from universities, consortia, hospitals, laboratories, all of that, and we will really kind of push them through all the EBI, the European Bioinformatics Institute's data resources, which are a number of them, and then make all of that, this kind of combination of processed raw, potentially raw data with added values, and make all of that available on a dedicated place called the COVID-19 data portal, which will then allow the users who may be clinicians, scientists, or actually policymakers to actually take advantage of what is happening, what is the latest changes or updates when it comes to research and our understanding of the virus. And as you can see, there were kind of two splits to this approach as well. So we had the open research data and access control data, because of course, as you realize, there were a lot of human data that we were also dealing with, and human data is sensitive and needed to be treated in a secure way to ensure the security of the individuals that are kind of whose data we were working on. So all of the human data will be in kind of dedicated data resources, which weren't publicly available to everybody. So we would ask for kind of access to those data, and everything else, all the viral sequences on all the isolate sequences, they are the data that are kind of public, right? So, and all of that will be was and still is publicly available. Now, during my time when I was part of the strategy office at EBI, what I learned were kind of, added, I guess, to what I had, picked up as a bioinformatician, but kind of, as I said, the direction of my path into this journey kind of diverted from programming. And the stuff that I learned during my time in the strategy office were multitudes, right? I learned how to write policies. I learned how to write proposals and white papers, medical strategies, and documents that would go to the governments. I learned how to coordinate and work with international organizations, researchers, scientists, and I also learned how to respond to requests for information from our partners, who could have been from any countries or any research institute across the globe. And I also learned how to work with large-scale data analysis. So these are not the genomics data. These are, kind of the behind the scenes of how many people have used the EBI's resources, for example. And then finally, after that time on the Wellcome Genome campus, where both the Sanger Institute and the EMBL EBI were placed, I moved into a new field, absolutely new field for me, so which is neuroscience and dementia. And I got this opportunity to, through the UK Dementia Research Institute, where I am based at the moment. Now, before we go into this, I want to highlight that dementia is actually it's a global issue. People living with dementia are increasing by numbers by year, and at the moment we have around 50 million people living with dementia across the whole globe. And as you can see, the number of people living in low and middle income countries are already higher than people living in high income countries. And this disparity will increase. This is what WHO has estimated by as the time goes by as well. So it is an absolutely crucial global issue that we all need to be aware of and do something about, along with all the other things that we all need to be aware of and do something about as well. Now, bringing it down to what we do and what you can do in the space of dementia. Now, keep in mind that I am not a neuroscientist. I am a bioinformatician. The field that I love is genomics. So for me, joining this organization gave me an opportunity to challenge myself, to learn new things, but also to see how I can adapt and adopt what I have learned to new fields as well. So this is kind of a plethora of tools and approaches that you can use when it comes to research in dementia. So it goes from omics data to neuroscience to imaging to stem cells to AI even. And what these translate to, for me, are different data types. And that's kind of what I am interested in and what I need to work with as well. And those are, again, genomics, transcriptomics, imaging, clinical data, healthcare records, behavioral data, where we are actually recording mice movements, for example. So what we need to do at UK Dementia Research Institute to kind of link all of these things together, right? And this is the strategy, the data science strategy that we have for the whole organization as well. And I'm very, very proud to be able to participate and deliver these as well. And those are 5 main principles. So data integration, how we link all these data types together. Data democratization, how do we make the data and the research that we are doing available to everybody else as well. Access and representation, where we actually make sure and facilitate accessibility, availability of our research, of our code and data to the entire population, but also make sure that the data that we are working with is representative of the global population, because as I mentioned, this is a global issue. How do we actually make sure that we offer training to both the UK DRI community and beyond as well, and make sure that we are sharing all of that and partnership? So I will just very quickly touch on a couple of these issues as well. So in terms of data integration, we are looking at knowledge graphs or graph networks of how you can link up different modalities of, for example, transcriptomics and proteomics, link them all together to actually create this much larger and more intricate image of how different data types and perhaps dementias are related to each other. And one of the ways we can then use this information is how do you go about and project informations that you have got from population at population level, for example, records, clinical records or health records, and project them to cohorts. Cohorts are basically a select number of individuals that have been approached or have volunteered to participate in research. And then how do we then link those to much smaller cohorts where there has been a specific, for example, people who live with Parkinson's have been approached where we have quite a deep knowledge about their genetics. Now, what I want to mention here is that many of the cohort data sets that we have are amazing. There are resources that are very, very valuable to the scientist. However, majority of them are not really representing of the entire population, again, of not even the UK or the country that they are based at. And this is something that I very much have made part of our data science strategy, is how do we ensure that what we are doing is actually applicable to people who may not have resources, who may not have access to our research. And in a data science way, the way we can do this is to ensure that the data sets, the cohort data that we are looking and working with, are the ones that are much more representative and much more diverse when it comes to ethnicity, for example, but also ensure that the people that are doing the science, that are actually learning how to do bioinformatics, are also part of this tapestry, right? And for that, at UKDRI, we have partnered with an organization called Into Science UK, where they place high school young people across the whole organizations, where they can actually get an opportunity to trial their hands on any part of the research that goes on. And for us, for the team that I'm part of, this is also something that we are planning to do. Now, finally, this is how I see the global world. Like, this is, I see it, okay, very, very nerdy, data points and networks, right? This is actually an image of the whole world where publications have been linked together, where they have kind of referenced, you know, each other's papers. And the thing is, it's a very beautiful image of just to show how we are all connected together as well. And I very much like to see this you know, concentrations of this image, which is again based on publications and is kind of mainly in Europe, to actually a bit more dispersed across, you know, the whole globe, because we do have amazing scientists everywhere as well. So the journey so far, okay, the top picture is like, that's your goal. That's what you're aiming for. You think you're just going to ride your bike. It's a straight line, but no, there are many, many pits. There are, you know, you have to climb, you have to swim. And this is not something that would be new to any of you. And just keep in mind that this does exist. And my suggestion to everybody here is actually do look up and do make sure that, you know, that glass ceiling, which will be there for you as an underrepresented data scientist, bioinformatician, that you make sure that you do break that. You do break that, and you do that by supporting your next colleague or the next student or the next generation of female bioinformaticians and data scientists that are coming, because we don't do, science is not done in silo, and it is quite important. Do ask questions and not be intimidated by perhaps others who may say, oh, this is a silly question. There are no such things. And then finally, surround yourself with your Avengers, and absolutely surround yourself with people who support you, who cheer you up, and will take you further. And they may be your friends, they may be your colleagues, they may be your family, but surround yourself with those people and take them with you on your journey as well. And I will stop there. Thank you very much. Oh, actually, I should just say, if anybody wants to get in touch with me, please do so. LinkedIn is probably the best place to reach out to me. Thanks very much. I'll stop sharing.
Thanks so much, Amonida. That is really, really amazing. Amazing journey. I really, really love that. I know that a lot of people have questions to ask. Please, I would like Amonida and Ruth to take the question at the end of the webinar after Ruth's presentation. So if you have any question, please drop it in the chat. During the question and answer section, you will also be able to speak. We will allow you to talk. So Amonida, thanks so much. That was great. That was amazing. I have questions too. So when the time comes for question, definitely going to ask. So let's go over to Ruth, and so that she can share a story. I'm going to give a brief profile of Ruth. So just give me a little movement so that I'm going to share a picture so that you can see the person I'm talking about. So Ruth is actually a computational Yes, she's a computational biology student from Oxford University. She's actually doing a PhD in molecular and cellular medicine at the University of Oxford. She is a scholar of Akfos Kennedy Trust Prize studentship, and she is actually the founder of My Science Journey. Maybe one of you, any of you have come across that platform or you've been following that platform. It's an amazing platform, and I'm very sure she's going to tell you a little bit about that platform today. And she is actually an amazing leader shaping impactful initiative across the globe. So Ruth is going to tell her a story and encourage us. I'm very sure you are going to be inspired. So Ruth, please, you have the floor.
Thank you. So I'm just gonna share my screen.
Yes, I'm going to stop sharing.
Can you see my screen?
Yes, Lucy.
Yes.
All right. Thank you so much. So hi everyone. It's a pleasure to be here. Thank you so much to Toby and the Bioinform her team for inviting me here. I'm always, glad to share my story and to, pay it forward. And I think this also is the reason why I started my science journey, because I believe that, every individual has a different story, and it's important for people to tell their own narratives. And then, in this way, you can pick one or two things, because everyone's journey is different anyways. So yeah, I'm glad to be here. I'll start. So I like to define myself in different ways. So I say I'm a computational biologist because I do a bit of bioinformatics, a bit of statistical genetics, a bit of computational things here and there. And I'm also a teacher. I have taught a lot of, especially bioinformatics modules. I'll talk about them in a few. And also I'm a mentor to a few individuals. So yeah, I also talk about my science journey in a bit. And I think the host has already mentioned that I'm currently pursuing my PhD at the University of Oxford. So I'm gonna start this slide. I am from Kenya. So those of you who don't know where Kenya is, this is where Kenya is. East Africa, close to the Indian Ocean. If you haven't visited Kenya, then this is your cue, please. Make sure you visit Kenya. And then a bit of fun facts about me. I just don't do science alone. I love outdoor as well. So these are pictures of me hiking in Kenya and also in South Africa. And recently I did a hike in North Wales. And yeah, so jumping in straight to my background. So this is just a summary of my undergraduate life. I started in the wet lab. My undergraduate studies was microbiology. Actually, initially when I was in high school, I had two passions. So I knew I wanted to pursue a career in medical sciences or medicine, for that matter, or pursue computer science. So I chose to pursue the medical science angle and decided to focus on microbiology, which I did at Pune University. And this picture here shows the bit of lab work, wet lab that I did during my internship. And then these are a few of my classmates. This here, let me get my pointer. Laser pointer. Okay, perfect. So this here is my very first presentation, actually. We had a symposium at Pune University, and I was presenting my research. And then this is my graduation, and this is my master's, my undergraduate supervisor here, very amazing individual who I am very privileged to have been mentored with. He's still my mentor until now. And I'll talk about the importance of that in a bit as well. So after finishing my undergraduate studies, actually before finishing it, I realized, I discovered that there's this field, that combines both my passion, like I mentioned, I was interested in medicine and I was also interested in computer science. So I realized there's bioinformatics, which sort of combines both computer science, statistics, mathematics, and the biology into one field. So I said, why not give it a try? So while I was in my 4th year, I, applied for this master's scholarship. It's called the Eastern African Network for Bioinformatics Training. Right now, they no longer run it, but it was a very good program while it lasted. I was part of the 2nd cohort. Presently, they still have the Masters in Bioinformatics at one university, but they are funded by DAD fellowship. So in case you're interested in pursuing a Masters in Bioinformatics, one university is, okay, I don't want to, so that I don't sound, anyways, one university is one of the best places to do your Masters. So if you want to pursue, then you can check it out. They currently have fellowships that are offered by DAD, so you can check that out. I think they are running their first iteration this year. Yeah, so I did my master's at one university still, and then the program was for 2 years. So the first year was the coursework phase, where we were taught technical bioinformatics skills. We did both the theoretical and the practical aspects. And the thing about bioinformatics is that it's very diverse. So you can take whichever angle that you want to take. If you want to focus on human research, if you want to focus on plant research, if you want to focus on animal research, if you want to look at the genomics aspect, if you want to look at the microbiome, there are different ways you can do bioinformatics. So I knew that, I'm passionate about the medical field. So I knew that I want to do bioinformatics that is applicable to the medical field. And for this case, I chose the human genetics space. So for my 2nd year, during my master's, I was posted to the University of Cape Town, where I did a research project trying to assess the accuracy of HLA imputation in African populations. So this also shows a bit of my master's journey. These are my wonderful supervisors from Cape Town. This is Nikki Mulder. This is Mamana Mbiavanga. And then this is my supervisor from Pwani, Suheila Hashim. I also had another one, Santi De Villas, but I didn't get her picture. But she was also very instrumental in my master's journey. This is my class before, while we were starting our master's program, the 6 of us. And then this is us after graduation. Here we've been combined with the immunology class. But yeah, some of us are here. So this is the before and this is the after. And then these 2 pictures here show the people that were also instrumental during my research phase. So this is Dr. Caleb Kibet. So probably some of you have heard of him or have met him, I don't know. And this is Winfred, who was also a PhD student. We worked very closely together. Eric, Pauline. I would just like to acknowledge these people because they also played a huge role in my master's degree. So in case you have any questions regarding this, I'm happy to answer them later on. But this was my master's phase. So like I had mentioned, I applied when I was still I applied when I was joining 4th year. So by the time I was finishing my undergraduate, I had already secured the scholarship. So I sort of like moved or jumped from the undergraduate to the master's. I actually graduated in between the master's for my undergraduate program. So yeah, it was a bit challenging when I was starting. Actually, not a bit. It was a lot challenging because imagine you've just been doing the wet lab aspect. You've never done the computational thing. And now you're in a class where you have to, learn all these things at a go. Sometimes you're trying to think, to make sense of the code, but sometimes things don't, make sense when you're starting out. So if this is you, just know that, it's a journey, and one day you're going to get to a place where you're like, you can even teach others, which is the place that I got to. So when you start, this could be challenging, especially if you're not coming from a computer science background or a statistics angle. It could be a bit challenging. But also if you're coming from computer science and statistics, you don't have the biological knowledge as well. So the biology might be challenging as well. So it's up to you to, take up the challenge and make sure you make the best out of it. So yeah, during and after my master's, I did a lot of teaching. So because the scholarship I mentioned was part of the Pan-African Bioinformatics Network, which is H3 Bionet. Probably some of you have heard of it. And H3 sorry. Okay. H3 Bionet runs a couple of trainings. There is an introduction to bioinformatics, 16S RNA intermediate bioinformatics training. So all these courses, I was a teaching assistant for the Kenyan class, and at one point I also was a teaching assistant for the South African class. And then during this phase, I also got trained as a carpentry instructor, and I got certified. So I also got to teach a few of the carpentry's lessons in Kenya and in South Africa as well. And then after my master's, I joined now a new program, which I will talk about in a few. But also while doing this particular program, I also was part of the residential training course, which is a course that is sort of like in the midst of you transitioning from the coursework to the project phase when you're doing the master's program in Pwani. So they usually have this phase where you do a residential training course, and then you get to work on a project while you're both together, because ENBIT was getting students from Uganda, Kenya, and Tanzania, and they were working from different places. So we had a class in Makerere and we had a class in Pwani. So during the residential training course, they will bring us together to work on a project together. So once I finished my master's, now I became a teacher for this particular class for the next cohort. And I also did this, which is what I want to talk about, which is, it was called the Bioinformatics Mentorship and Incubation Program. But basically what it was is that it's an internship program. We took interns, so people that have completed their undergraduates and are interested in pursuing bioinformatics. So we'll train them for 6 weeks at ISIPE. ISIPE is International Center for Insect Physiology and Ecology in Kenya. So we'll train them the bioinformatics aspect. In the first 2 weeks or 3 weeks, we will teach them basic bioinformatics, and then in the last phase of the internship, would assign them mini projects and ensure that we pair them to mentors so that they also get the feel of, not only the theoretical, but the practical aspect of things. So this project has also been published. You can read more about it. This is the paper here. And before I am crucified, I just got a few pictures of my interns. I couldn't trace the other ones, but these are some of them. This is one cohort and this is another cohort. But I think we ran 6 cohorts, but I assisted with 3 cohorts, and then I ran 3 cohorts. And this is my supervisor here at ISIPE, Dr. Caleb Kibet. This is the PI for the project, who's also my supervisor and was also a very wonderful mentor. He's called Dr. Daniel Masiga. And then this was our project manager, very wonderful human being as well, Karen. And these people were very instrumental in my time at ISIPE while I was doing this program. Unfortunately, it's no longer running because, like, with most science programs or projects, funding comes to an end. And for this case, the funding came to an end. So it's no longer in place, but we hope that it can come back, because a lot of individuals that went through this program, I think I saw some of them join the meeting. A lot of them that went through this program were able to secure master's scholarships and even job opportunities, both in Kenya and, all over the globe. So it was, a very important step towards building their career in the bioinformatics space. So yeah, maybe people all over, the continent can pick this up as well and try to run it in different parts of the I was about to say country, in different parts of the continent, the African continent, because I believe it was a very useful phase for most people, because it's very short. so it doesn't consume a lot of time, but it also gives you an idea of what to expect in the bioinformatics space, so that you get to explore and say, okay, this is something I'm interested in, let me pursue it. This is something that I can't do, let me find another, path. So yeah, this is what I did right before I joined Oxford for my PhD. And again, I started applying for my PhD immediately I completed my master's. So when I was running this program at some point, I knew that I was leaving for Oxford at some point. So yeah, I think I would want to also just acknowledge the fact that I don't want to say that things just worked out for me, but I also got a bit of the normal things. You're applying for something, you don't get it, you keep trying, you don't get it, but then just one day or everything falls in place. So if you feel like you're trying to apply for programs, trying to apply for opportunities and they're not working out, it's just probably because they're not the right program for you. So keep trying, and then you might end up in something that you actually like. Because when I think about my journey, if I had given up and not continued applying, then it meant that I'll probably not be in Oxford right now. So yeah, so keep on applying and you might end up in a position or an opportunity which combines both your enthusiasm, passion, and your career trajectory. Okay, I think I'm speaking a lot, so I'm gonna turn it down. Yeah, so now this is me doing my PhD in Oxford. This also summarizes my journey. I don't even know where to start, because I've just done 2 years now, and I feel like I've been able to engage in so many activities which I'm so grateful for. But yeah, this is my wonderful lab at Oxford University. This is my supervisor here. And then these are my team members. This is Dr. Carol Ibe. She was very instrumental in pushing me towards applying to Oxford. And then this is my matriculation picture. So in Oxford, you have to matriculate before you start your studies. And then I got to do a short internship at Harvard Medical School sometime this year. And I got to meet King Charles at Buckingham Palace. And this is me and my mentor as well, Dr. Anne McKenna at Buckingham Palace. And I got to serve in the wonderful African Society in Oxford as the vice president, and we were able to convene one of the largest African-focused events in Europe, which is the Oxford Africa Conference. And we hosted, different global leaders from across the continent. But here I highlight Raila Odinga, who's a prominent leader in Kenya, because I'm Kenyan and most of you might relate with that. But yeah, the PhD journey has been amazing. I mean, I still do bioinformatics, but now I do more of statistical genetics as well. My research is centered around understanding disease association in the context of HLA as well. So HLA is the human leukocyte antigen region. It's part of our immune system, and it plays a huge role in antigen presentation. So let's just say my research is centered around understanding disease association, and I use statistical genetics approaches and also bioinformatics approaches to make sense of this. So in as much as I'm doing a degree in molecular and cellular medicine, I'm purely computational, so I don't do any wet lab. And that is the thing that I love about my research, because once I said I'm doing bioinformatics, I was like, no going back to the wet lab. So I am so happy that I got to do a project that I actually wanted to. Not to say that wet lab is not good. I mean, if it's something you like, please go ahead with it. But I just didn't like being in the lab. I liked being behind a computer screen, a black screen, running codes. So that is what I was passionate about, and that is what I chose to pursue. So if you're passionate about, both, because I've met a few bioinformaticians that also do the wet lab aspect, you can do that as well. So that's still okay. Yeah, and also during my time, my 2 years in Oxford, I've also done a bit of teaching. So I think probably some of you might have heard of or might have done it actually. We just ran the second iteration of the Bioinformatics for Biologists, analyzing and interpreting datasets. This is run on the FutureLearn platform, And this time around, we had 15,000 people enrolled. So probably some of you did it. And it's hosted by the Welcome Connecting Science. So I'm an instructor and a course developer for this particular course. And I also got to do a bit of teaching assistant as well for the genomic medicine course in, for the Masters in Genomic Medicine course in Oxford as well. This is my first here. And I'm also the carpentries liaison for the Reproducible Research Oxford team in Oxford as well. So this is to just show you that, you know, as you can remember, like I mentioned, I started from the point where I moved straight from the wet lab to the dry lab. No computer science background, no, you know, statistics. I mean, we did a bit of statistics in undergraduate, but if you're doing a microbiology, biochemistry, you know, they pump more biology or chemistry rather than the statistics or the whatever computer science angle. So this is me starting, you know, in a field that I was very new at, and I'm just trying to show you the growth that I have had from that capacity to, you know, leading teaching programs, developing courses, you know, and participating in all these spaces. So it means that, you know, once you put your mind to something, you can actually do it. So this is to just show you that, you know, if I was able to do it, you can also do it as long as you're passionate about this field. All right, so jumping into my son's journey. This is a project that I hold dear to my heart because, like I mentioned, it's important for people to give their own narratives. You know, most of the times we might just say, you know, so and so did this and this, but once they're able to tell their story and personalize their experiences, then you get to relate or to get to understand that, you know, whatever you're going through is just a phase in the journey, and that it's not unique to you. It's something that happens to everyone. And, you know, it's just about what you make of it that will determine where you end up. So what we do at MSJ is that we try to bridge the gap between experienced scientists and emerging talents. So it's sort of like an intergenerational platform between students that are starting out, so undergrads, postgrads, and then we have senior scientists, PIs, people that are in industry, people that are in academia, people that are teaching. So we host different people in the science space. And, you know, if you haven't heard of this before, this is your chance to, you know, get to join us, and so that you can get to understand or get to network with these people. So we not only give them the space to share their narratives, but we also give you an opportunity to meet people and, you know, to form collaboration opportunities and to network with them. And also, you know, you never know, you can get a mentor from one of these individuals. So that is what we do. This is the team here. Probably some of you know Anita Makori because she's big on LinkedIn. And then we have Cynthia and Nehemiah as well. And this is our first industrial visit. So we're able to visit one of the largest pharmaceutical industries in the world, Novo Nordisk. They have a branch in Oxford. So some of us got to visit that. So this is what I'm talking about. You know, we just get to do the bit of, you know, introduction here and there, and then from there you can pick it up and build your own story. So we've hosted 34 speakers from all over the globe. So we've had people from Kenya, Nigeria, UK, US, Australia, Nigeria, Uganda, Ghana, Gambia, South Africa. Some of you might know some of them. All this information is in our website. So you can just go click on our website and look at all the speakers we've hosted and what programs that we are planning to do, or whoever we are planning to host. And, you know, the thing I tell about people is that science can be done in different ways. You don't necessarily have to be, you know, go from master's, PhD, become a PI, you know. You can go to master's, join industry, master's, PhD, join industry, master's, start, you know, a program that trains people, like do what the Welcome Connecting Science does, you know. So there's different ways you can do science, or start something like Africa Oxford Initiative, where you get to support African researchers in Oxford. So you can start that in another institution. So, you know, like there's the baseline, yeah? You did an undergraduate in science. You know, you want to do bioinformatics, yeah? You can do it. If it's something you're passionate about, you can continue with the path, maybe venture into industry, venture into academia. If If not, you can go and train people, you know. You don't have to stick to a particular path. That is what I'm trying to say. And that is the stories that we tell on MSJ. So these are our social media platforms. Make sure you follow us. This is the website I was talking about. You can browse it and see what we do. We've recorded the sessions and have them available on YouTube, the ones that we've had previously. I haven't uploaded all of them yet, but some of them. So you can check them out and get to listen to these stories. And probably you can relate. Actually, not probably. I can assure you, you will relate to, the stories that are being told. And if you're, in case you're interested to, speak to one of our speakers, just let me know, and then we can do the, connection. You never know where these things go to. So yeah, these are our platforms. You can find us there. And just to finish with the tips that I have picked, I am still learning, by the way. I haven't perfected this. I'm also still learning. And the thing I've come to realize about this science path in the bioinformatics space where I have contributed a lot is that your network is your network. A lot of opportunities that are available, can easily be accessed through your networks. You don't even need to look further, or let me go and look for. So if you put in the work and show people that you can actually do something, they will just send you an email, Ruth, I think you can do this. Why not apply for it? Or you'd reach out to someone and tell them, I have been working with you on this project, but I want to do this. What do you think? Can you recommend me to someone? You understand? So make sure you capitalize on your networks. And that is how you get to build, a network of people, because this one will refer you to this one will refer you to this one, and you get to know a broad range of people who might come and assist you, because science is not a one-man show. I think Amonida has already spoken about that. You have to work in collaboration. So, if you're able to collaborate and work together with different individuals, then it means that you get a better chance of, succeeding or whatever career path that you choose to focus on, whether in academia or in industry. And also maximize on available training opportunities and mentorship opportunities. We are in an era where everything is available, like technologically available. You cannot say you didn't know, because everything is online. You just have to go online and search. HackBio, for instance, I think Bioinform does the recommendation, but HackBio runs bioinformatics opportunities. The Welcome Connecting Science, which I was part of, also does bioinformatics trainings. Omics Codathon does bioinformatics projects as well. So there's I'm just mentioning a few, but there's a broad range of things that you could do to build on your skill set. There's also a wide range of mentorship opportunities. I mentioned Carol Eve because she was instrumental in my journey to Oxford, but I applied into her mentorship scheme. She just didn't pick me and say, come, I want to mentor you. So you have to reach out to people. You have to reach out for your opportunities. Like we're running MSJ, you get to join a session and you figure out that this is someone that can mentor me. You reach out and say, I want to talk to so-and-so. Can you please link me up? You understand? And also, the other thing is that your soft skills are also very as important as your technical skill. So in as much as you're building your bioinformatics programming skills or your biological skills, make sure you also build your soft skills, how to communicate, how to present your work, how to speak to people, how to collaborate, So make sure you also build on this, because a lot of times people can just, brush you off because because you haven't built on your soft skills. Sometimes you don't even need to be very good at something. You just need to show people that you are willing to learn, you have the willingness to take a training opportunity, you are something that can unlearn to learn, and then they would have confidence and say, why not just try her? We can train her for 1 month and she can pick it up from there. So soft skills are very important. And then the other thing I like to talk about is, I think I've also mentioned this, that focus on your passion. It's not worth spending 20 years doing something that you don't like. Imagine waking up in the morning, you're stressed. Now I have to go to that office. now I have to do this work. I don't even, I don't even understand it. And I think about the thing about bioinformatics in that is that, or the science space is that, you might want to just do it as like everyone is doing. Oh, I want to do a master's, I want to do a PhD. But if you're not passionate about it, you'll be stressed. For instance, a PhD is 4 years. Imagine 4 years doing something you don't like, working with the supervisor you don't like. Imagine how stressful that would be, especially right now that we have a lot of mental health issues. So yeah, try to focus on something that is you're passionate about, something that, if someone wakes you at midnight, like, Ruth, can you do this? Then you say, yeah, let me do it, and then everyone's journey is different. I think you need to also get to understand that if you start comparing yourself, oh, I want to be like Ruth, or I want to be like Amonida, I beg, you're it's not gonna work. Like, I think they're saying that comparison is a thief of joy. And you build your own journey, build your own story, get to learn from people's story, learn their mistakes, learn their achievements, but use that to build your own. Don't say, okay, now because Ruth applied to do a PhD in Oxford, now I want to do a PhD in Oxford. No, you might want to do a program that is not offered in Oxford, right? Not just because you want to end up in Oxford, you also want to do something that you like, something you're passionate about. You want to work with someone that would actually mentor you properly. So if you get that person in London School of Hygiene and Tropical Medicine, go to London School of Hygiene and Tropical Medicine. You don't have to come to Oxford. So yeah, basically I'm just trying to say that your own journey is different, and then try to give 100% to everything you do. Like I mentioned before, people watch. Sometimes you think people are not looking or watching, but people actually do look at what you're doing. And, this is where they can say, hey, by the way, I saw Ruth present her research, at this convention. What do you think? Maybe we can invite her to come and give us a talk. You understand? Or they can say, maybe we can invite her to come and teach our students, So you have to show them that, I am able to do this, so that when you're applying for opportunities or for whatever, people don't have to look too hard to try and understand, oh, okay, why do we need to take this person again? Because your work already speaks for yourself. Your work is already out there. So and so can clearly give a statement about you. It doesn't just have to be the normal where you choose someone as a referee and that's the only reference you get. Your work should also speak for you. People should also speak for you. So make sure you give 100% so that someone can confidently, because I don't think there's anyone that wants to put their name into something that is not working. So if you're giving 100%, then everyone will be confident and say that, I can bank on Ruth, I am sure that if I give her this task, she would do it. And then I've talked about collaboration, and then there's the other aspect where rejection is redirection, my friends. Don't allow rejection to demoralize you or to bring you down. If you're applying for something and it's not working, if you're trying to do something, and especially for bioinformatics, especially if you're starting, sometimes the code is not working, or this program you're applying is not working. Don't force it. Try something else. So sometimes, you don't know until you keep trying, and then you end up at some point, and then you start thinking, by the way, that thing didn't work out, but it actually helped me, because if I was doing that, then probably I would have failed. You understand? So yeah, and then finally, pay it forward. Like the way Bioinform is doing to try and, inform you about the bioinformatics space, offer modules and whatever contribution you can give in whatever, whether big, whether small, it's very valid. Make sure you also pay it forward so that those that come after you don't have to have it, don't have to do it the hard way. They have to at least get to do it much softer than how you found it. So yeah, try to pay it forward as well, and ensure that we have more women in these spaces, because a lot of times we say that they're not doing this, they're not doing this, they're not doing that, but it's because you don't have a seat at the table, right? So make sure that, when you're paying it for, then you're giving an opportunity for a lot of us to come to the table. So we will be in spaces where we don't have to keep complaining and say, there is no representation. Because if someone is looking to get maybe Black individuals that want to do this program, they don't have to look further. They can just say, let's take these 10 people from here. Let's take these 20 people from here. And it's because these 20 people are available. So you can't also work with something that is not there. But for you to work with something that is there, then it means you have to have built that, you know, cohort or group of individuals. So try to also mentor others and ensure that there's a lot of us in these spaces so that we can take up the space that we need to take up. Yeah, thank you so much for listening to me. These are my socials. More active on LinkedIn, so just send me a DM if you would like to speak to me further. Follow me on both Twitter and LinkedIn. And yeah, I'm happy to answer or take any questions.
Yeah, thanks so much. That was wow. Thank you very, very much. And yes, we can have questions now, just as you said. Our time is well spent, so we have just 15 minutes. So if you have any question, you are free to indicate by raising up your hand. And we take like, I think we should take like 2 questions, or yeah, 2 questions. Then Amonida go first and answer, then Ruth go Ruth go second. But we have just 15 minutes, so we won't be able to take much question. And yeah, but you can always reach out to either of them via LinkedIn or any other platform if we do not answer your question today. So let's start. Please, if you have any question, you can indicate by raising up your hands. Okay. Okay, so let me see, please. Olatunji, am I right? I'm coming. Okay, Marvin, please, you can speak. And please let us know who you're directing your question to so that they can answer it properly. Yes, go on.
Okay. Hello, everyone. My name is Melvin Hakabo. I'm from Nairobi, Kenya, and I'm currently doing the HackBio internship under the Bioinform Scholarship. So my question is directed to Amonida, because your journey is really interesting because of the certain parts that you've gone through that to some of us look like very big, ambiguous goals. So my question is, what's your advice on securing graduate research programs in bioinformatics if you have little to no experience at the undergraduate level?
Well, first of all, I just want to say again, thank you very much for the organizers, and also what an inspiring presentation, Ruth. That was absolutely, yeah, I'm full of awe, and it's wonderful. It's genuinely wonderful to see that what you have achieved. Melvin, thank you very much for your question. So, I have to say, and I will start by saying that I have had and still do have a lot of privileges because, as I mentioned, I have it may not sound very like a big thing, but I do have a European citizenship. And that actually opens many doors for me, which are not open to a majority of people who, on this planet, but also majority of people who would like to do bioinformatics, especially if you're an underrepresented individual like yourselves as well. So it is, I would just kind of echo what Ruth was saying in the terms that nothing is impossible, but you need to have some level of resilience and not give up. But in addition to that, I think again, you need to be able to actually achieve what you want to do in a space that is supportive of you, right? If you don't have access to opportunities, to activities, then it becomes quite difficult to be able to do that. So there are programs, Melvin, that do allow a master's. I think Ruth's examples were wonderful there, where you can actually enter the field of bioinformatics and data science without any prior knowledge, and that's absolutely possible. I have had many colleagues, all based in the UK, who came from a pure biology background and they had finished their PhD, but they wanted to enter in bioinformatics, and they did a one-year master's, for example, and they then continued working as bioinformaticians, very successful. So we'll refer back to the suggestions that Ruth had made. I think there were wonderful, very good practical examples of how you can approach and how the programs that are available as well. I would also say, if you don't perhaps are aware of opportunities, do reach out to people that seem, for example, on LinkedIn, who seem to be aware, who seem to be active in these spaces, and do reach out to them and ask them as well. Be mindful that they are doing other things and they're not just, kind of checking their LinkedIn profiles as well, like Ruth here. So be mindful of their time, but do reach out to people who seem to be open to support you. I'm very sorry, it was not very hands-on example that I can give you, but I think the hands-on example actually came from Ruth, which I think they were very good. I hope that answered your questions that you had, Melvin. Ruth, would you like to add some stuff? I think you had more concrete example than I had.
No worries, Amonida. I think you just gave it, the perfect response, and I don't want to circle back to the things that I've spoken about, but just to echo you that, you have to go for the opportunities, because hello, you're a Black person, a Black woman. So yeah, it's gonna be a bit chaotic trying to traverse that journey, but it's possible. It can be done. And I think a lot of us, I'm just a representation, but a lot of us are in these spaces. So it just shows you that it can actually be done. And maybe to speak about, the getting into bioinformatics from a wet lab perspective, like I mentioned before, it's are you passionate about bioinformatics in the first instance? Yeah. Is it something that you just want to do for the sake of doing it, or it's something you're passionate about? If you're passionate about it, then it means you're willing to learn. If you're willing to learn, then it means you're willing to take up training opportunities like HackBio or Mixcodathon, Welcome Connecting Science. This is where you start from. And then by the time you apply for a master's, people actually see that you're actually passionate about this, and they can give you an opportunity to start it. I think that is what I would say for now. Thank you.
Okay, thanks so much for the response. So we have Uto Bonk. Please, can you go ahead with your question?
Hi, it is 11 a.m. in Nigeria, 11:21. So good morning, everybody. And thank you so much for this webinar. It was really insightful. So my question goes to either of them. Anybody that can answer it, that's fine. I'm not really directing the question to anybody, because both stories are like kind of diverse. So this is it. I'm quite interested in bioinformatics, but then I have like found a niche, just like Ruth said, that bioinformatics is really broad and you have to like pick a particular part of it and then focus on that and then build your space, because it's really broad and quite noisy if you do not know what you're doing per se. And what I have actually found also, I got your internship, but I'm not really the genetics person. I'm more into drug discovery. That's where I want to like build my own niche. That's where I want to grow career-wise. But the problem I have at the moment is like mentorship, and Ruth happened to say something that was like in that line. So I have done like a computer-aided drug design courses. I've taken a computer-aided drug design course, but I haven't like really had internship opportunities or opportunities for like mentorship. I haven't really gotten a mentor. So Ruth, this is to you. Do you have anybody in your space that I can be recommended to? Or do I need to like reach out to you? I'm like shooting my shots publicly. Yeah, thank you.
I like that.
Yeah. Also depends with where are you open to like doing it anywhere in the globe, or you have a specific place you want to do it? Because also Amonida has been to these spaces where bioinformatics has been done, like Embol ABI. You know, I've been at the Welcome Genome campus, by the way, Amonida. And I think the thing about these spaces is that also drug discovery is broad. You know, you also need to figure out or know which space of drug discovery you want to do. Do you want to do like the molecular docking stuff in bioinformatics?
Yeah, more like it.
Yeah, so maybe we can have a conversation later on. But I think also Amonida might have interacted or come across individuals that are doing
Like,
this.
I'm open to anyone, so I can reach out to any of you, right?
Yeah, absolutely. Please do, and that goes for everybody. I mean, I wish I could offer mentorship to everybody who enters in this space, but unfortunately, my time is also limited. But I am planning to put kind of together a set of resources, because I do get approached by many people asking these very, very important questions. And I'm hoping that I can kind of collate all of that and put them on my LinkedIn somewhere, which will be useful. But yeah, please do reach out to myself and Ruth. In addition to that, also think about opportunities that may not necessarily be at a university or academia. Ruth may highlight that as well. Industry. So there are a lot of companies who actually offer internships, which may not be exactly mentorships, but an internship venturing into, you know, for example, bioinformatics or drug discovery. I mean, drug discovery itself is just huge. You can do drug screening, you can do compound screening, you can use AI for all of these things as well. So it is a huge space. So do look up the big companies who are within that space, and majority of big companies do have a very strong bioinformatics program and also research, and many of them also actually do again internships as well, which could be very, very helpful.
Thank you. I think that answers your question, Tombong, right?
Yes, it does. Thank you. It was really useful. Thank you.
Okay. I don't know if there's anyone that have a question again before I
think there's a question around a PhD in bioinformatics. I think I've seen two people ask about that.
Yes. I wanted to like let people speak first. If there's no one who is ready to speak, then we just go to the chat and pick the question. Okay. But since there is none, let's just start from there. The question is about what are some tips for applying for PhD in bioinformatics or computational biology? Are there certain things or a certain level that they expect from a PhD applicant? So I know both of you are, okay, Amonida's past that stage, but she's been there and Ruth is currently there. So anybody can answer?
Ruth, please go ahead.
Okay.
Okay, a PhD in bioinformatics. Okay, so the thing about most of the universities is that most of them don't have a PhD tagged PhD in bioinformatics. So that's one thing you need to understand. A few of them do, but most of them don't. So like, for example, for my case, my PhD is called molecular and cellular medicine. You might just see that and be like, I'm not doing it, right? So the fundamental aspect or the thing that you need to know is that what kind of research do I want to do, right? For example, I want to do drug discovery, right? So I am looking out for projects or for PIs that are doing drug discovery. These PIs might be placed in programs that are called differently. So they might be called, PhD in statistical statistics, or PhD in molecular medicine, or, it might be termed differently. And I think that's what I've realized about the schools in the UK. And I think some of them in the US as well. In Africa, maybe there's one in Cape Town, in South Africa, that calls it PhD in bioinformatics. But the thing is that don't just look out for PhD bioinformatics. Look out for projects that have the bioinformatics angle. Look out for research projects that have things that you're interested in. And then these are things you can discuss with your supervisor. So in case you see someone doing drug discovery, they're probably a wet lab PI. You come to them and say, this is what I want to do. We can get you a second mentor that has the computational angle and will assist you, but I'll still be working with your group. So don't narrow your search to just PhD in bioinformatics. So I hope that answers that question.
Yes, that answers it perfectly. And I want to give an example too. For instance, I'm doing my master's. I'm actually doing my master's in immunology, but literally I'm doing bioinformatics. I'm actually working on spatial transcriptomy. So I've been doing a lot of coding, a lot of analysis. So yeah, just as Ruth said, you don't look at the name, look at the projects you want to do. What are you interested in? Then talk to the PI. This is the project I'm interested in. Can we bring bioinformatics into it? Yeah, thanks so much for that. Okay, there is someone that raised up our hand. Nuhela, do you want to go ahead?
Yes. Thank you, Ruth and Amonida, for the wonderful presentation. I hope you can hear me. My network is a bit unstable, but my question was on AI. I heard Amonida mention something on AI and drug discovery. So I need to know, with the rapid advancements that have been happening in AI, how have you coped with that? Because in recent ages, we used to learn codes and everything, but right now we even troubleshoot using ChatGPT and other models. So how have you coped with that? Do you think that you're going to be wiped out of the job market because of AI and machine learning? So that's that.
Thanks very much for that question. Well, first of all, I'd say we haven't coped yet. we are learning. I think that's true for all of us. I guess at UK Dementia Research Institute, we do actually have a very strong research component, which is pretty much entirely machine learning and AI centric. And that's where they look at for example, sleep mats. They collect data from sleep mats that they have put in people who live with dementia in their homes, and they collect the data and they analyze all of that data and making sense out of it by just using machine learning. So we are still, as a research organization, I think we're still kind of in life sciences, I suppose, in the beginning of how we can actually take advantage of AI and machine learning. There is and it will absolutely impact everything. And I mean, we already know that AI is everywhere, right? But there are I'm quite excited about it, I have to say. I'm very excited that we can take this technology and make it much better. But I'm also quite cautious in terms of how we actually implement AI and machine learning, because there are a lot of issues around ethical aspects of AI and how it's being used and which data set are being used as a training data set as well, right? Because if you can imagine, if there have been devices, for example, that are used for detecting I think pulses, or taking people's pulses, but they don't work on skins that have got more pigmentations than the usual perhaps subject that the data tests or the training set was trained on. It means that, you know, these devices will not work on people who are Black, who are brown, who have got, you know, a different ethnicity perhaps. So there are other aspects of AI which are, again, you know, this is what I mentioned about like which data we are using, you know, how varied and diverse data sets are. That all of that kind of leads into the training models that we are creating to perhaps be skewed. So it's also very important to be mindful of these things, but at the same time, it is very, very exciting. So I don't really think, I hope not, I don't really think that AI will put me out of my job, because there needs to be a human element to make sense of what AI is kind of, you know, giving back to us as well. And it will take some time before, I think, in my opinion, an AI system can be as subjective perhaps, or objective, depends on how we look at it, as a human being. So I'm kind of very excited, but with the, you know, with some cautious.
Thanks so
Okay,
much.
thank you.
Yeah, we have run out of time. I'm so sorry if your question has not been answered. Could you please reach out to Ruth or Amonida on LinkedIn? And I'm very sure they will be so glad to answer your question. I'm very sorry, we've run out of time already. Okay, so I will hand over to Toby to say our vote of thanks. But from me to Amonida and Ruth, thanks so much. It was amazing having you here. It was really nice listening to your story. So over to you, Toby.
Hi, hello everyone. Thank you for taking our time to joining us. It's been a wonderful time. Thank you to Hamonida. Thank you to Ruth's very inspiring journeys. You said a lot of things that you wouldn't even find on your LinkedIn profile. So there's a lot more, and it's very, very inspiring and enjoyable. Thank you, and for the efforts you both have put out in ensuring bioinformatics is where it is, no matter how little. Yeah, and thank you for everyone that joined. I believe it has been a wonderful time for you as well. The meeting is being recorded and would be sure to make it available as soon as we can on our YouTube page. And then I just want to say thanks, as I've said, to everyone that has contributed to the growth of Bioinforma. It's been a wonderful journey. This is the second iteration of our webinar, and as I can see, it's getting better and even more inspiring. And we hope it to be more inspiring in the future editions to come. So just before we go, I will just seek that if everyone can switch on their video just for a second while we try to take a virtual group photo together. So if you can switch on your videos and then I take a screenshot of everyone from my end, we would really appreciate that. I hope you had a wonderful time. It was very nice to me. It was a nice conversation, lovely questions, lovely answers, lovely experiences, and which is what we hope to do. So thank you again to everyone that turned up. It was really nice meeting you all. Okay, yeah, I think I have some here. Yeah, okay.
Okay.
Yeah, I think I've tried to do that now. Now, wonderful. Yeah, so we'll let you guys know when the recorded version is available so you can listen to it again and then gather more insights. Thank you once again, and I hope you enjoy the rest of your day. All right.
Nice.
See you.
Can it